Hello, everyone, and welcome back to Genetics. We are going to be talking about DNA in great detail over the next few weeks, and I am really excited for this. We are going to be learning about DNA replication and all of the various proteins involved in it and the logistics of how a cell actually makes a duplicate copy of its DNA and how it makes sure that that duplicate copy is correct. We're going to get into some of the applications of things that you can do with DNA. And we've touched on that briefly in the past already with, DNA finger typing with the short tandem repeats and the single nucleotide polymorphisms. And we looked at gel electrophoresis. We're going to be getting a little bit more in depth into some of those things and more in this chapter. And it's only going to get better from here. We're starting off today's lecture just with a quick note. So far in the course, I've been going pretty steadily through the textbook, um, either a lecture per chapter or two lectures per chapter, actually, more like it. And I haven't really skipped much of anything, but I am skipping section 5.3 in the textbook. We're going right past that, and I do think, however, that it is information you need to know. So your assignment is to read section 5.3 because that that really is the one that I have not covered in any great detail. It's all about inversions and more about recombination and how recombination can get thoroughly messed up when you have homologous chromosomes lined up and some of the parts that are supposed to line up together don't. So that's very fun and interesting things to learn about. And if we were working with Drosophila in the labs live, then we would be covering that in great detail. But for the time being, I think we're going to just put that off on the shelf. It's definitely information that you need to read. I don't believe I'll be testing you on it, but I may include questions on it in problem sets. So read section 5.3. Now that said, there's a section in that on something called Pachytene chromosomes in Drosophila, and that's not something that you need to really concern with. That is really fun information, and it's a huge genetic resource in the laboratory, but in terms of just sheer knowledge, if you wind up working in a Drosophila lab, it'll be handy and important to know, but for now, it's not among the more important things that we're going to be covering. So read section 5.3. 5.4 and 5.5 have basically covered everything in previous lectures. So we are going to move on now to the nature of DNA and the nature of genomes. I'm done talking about chromosomes. They're not quite as fun as DNA and genomes in general. That's more just that's a logistical issue of how the DNA is kept in a location, what box it's stored in. And I'm not so much interested in that as much as what the DNA is doing and how it's responding to its environment and how does it exist in the first place. These are some great questions. The very first section of this chapter, chapter 6, feels a little out of place and is talking about complexity of DNA. I would have put this later in the book, more with the genomics issues, but let's get into it. Basically, from this slide and this little section of the book, all you need to take away is the number of chromosomes in an organism, the number of genes in an organism, the quantity of DNA in an organism has no bearing on its complexity as an organism. Now, a lot of people will say, the terms more evolved or less evolved organisms, but evolution is change over time, and I've got news for you. All organisms alive today have been evolving for the exact same amount of time. We all started from the same single-celled goop, and we are all here today. So we have all been evolving for 4.2 billion years. So no organism on this earth is more evolved or less evolved than any other organism still surviving. So that's not a good phrase to use. Don't use it. It's pseudoscience, and it's pop culture that makes no sense. But there are organisms that are more complex than others. And at the larger level, at the cellular level, we all have approximately similar levels of complexity. Certainly the eukaryotes are all similarly complex, and the prokaryotes are similarly complex with each other, and the viruses are similarly complex with each other. But I wouldn't say that a virus is as complex as a eukaryote. But the point being, the quantity of DNA in your genome is not a good match for your complexity. Let's compare, Drosophila and the salamander. If you look at this chart, Drosophila has 180,000 bases, so 180 million bases. And this particular salamander has 90 trillion bases. They're on vastly different chromosome numbers, 4 versus 14, and that makes sense, because how are you going to fit 90 trillion bases on 4 chromosomes? It's going to break up into larger chunks. But the point being, the salamander is not significantly more complex than a Drosophila. Now, because salamanders have so much DNA, their cell cycles are actually significantly longer. The typical mammalian cell cycle is about 24 hours. The salamander cell cycle needs 48 hours just for S phase. And then there's the rest of the cell cycle as well. So there's a lot of DNA that has to be replicated and error checked in organisms with larger genomes. And it really doesn't have all that big a change on gene number either. Now, if a organism is tetraploid instead of diploid, it's going to have twice as many genes as a diploid relative. So twice as many genes, but they're not different genes. They're just extra copies of the genes the organism already had. In that case, what usually winds up happening is the two copies, the one from chromosome 5 and the one from the duplicated chromosome 5, they wind up sort of each specifying into little sub-areas. If a gene originally was expressed both in the heart and the brain, and then the chromosome that gene is on duplicates, after a couple of thousand years, you might find that one of the copies is expressed in the brain and one of the copies is expressed in the heart, and not vice versa. Or they might break down their functions in other ways, or one might slightly change its function. But this is all to the side fun anecdotes. Let's talk about DNA in more specific details here. So this genome section is the part that I felt is interesting to include, but out of place in where it's placed in the text. So I'm bringing back this slide from earlier in the semester, and this is just a refresher of what a nucleotide is made of. So a nucleotide is basically 3 main components. You have your base, you have the sugar, and you have the phosphates. So the base and the sugar, the bond that holds them together is called the glycosidic bond. And if you think sugars are glycosides, or sugars are glucose and similar things, and the bases are nucleosides, the base and the sugar bond is the glycoside, the glycosidic bond. That's how you remember it. It's a combination of the two words. So the base and the sugar combined, in this image the blue and the yellow, together they form a nucleoside. That's with an S, nucleoside. The only difference between a nucleotide and a nucleoside is whether or not there are phosphates. So you have a base or a nucleoside, or a nucleotide, depending on how much is there. Now, the monophosphate, diphosphate, and triphosphates, all of those are nucleotides. So the words that I have circled here, nucleoside, monophosphate, the reason that says side instead of tide is because in that case, you're describing the nucleoside and the phosphate separately. So if you're describing the nucleoside apart from the phosphate, then it is a nucleoside. But if you are describing the nucleoside and the phosphate as one entity, then it's a nucleotide. And the mono, di, and tri obviously is if there's one phosphate, 2, or 3 attached. And that 1, 2, or 3 has enormous consequences for every aspect of the cell. Hopefully you will recall from any high school biology or Bio 1000 and 2000 that ATP is the driver of most of the cell cycle. It's all of the energetics of a cell, and in some cases GTP as well. And the cycling back and forth between ATP and ADP, well, ATP is adenine triphosphate, or adenosine triphosphate. GTP is guanine triphosphate. It's literally just the purine bases. So those are incredibly important, and whether they have 1, 2, or 3 phosphates dictates a lot of how they work. So let's look at this in terms of the double helix and DNA bonding. So the bases can be broken down into 2 main categories: the purines and the pyrimidines. Now, right off the bat, I will give you some hints on how to remember which are which. Pyrimidine is spelled with a Y, P-Y-R. The 2 pyrimidines are cytosine and thymine. They both have a Y in their spelling, C-Y-T-O and T-H-Y. The purines do not have a Y. The word purine doesn't, adenine doesn't, guanine doesn't. So just to remember in your head which are which, the pyrimidines, C and T, have Ys. The purines, A and G, do not. Now, that's a difference between their names, sure. That's not going to tell you any difference in their structure. The main point to tell apart the purines and the pyrimidines is the pyrimidines have a 6-membered ring. It's 4 carbons and 2 nitrogens. The purines have 2 rings. They have that 6-membered ring, and that is bound to a 5-member ring. And you can see that in the diagrams here. Now, in both cases, the ribose or the deoxyribose sugars bind to a nitrogen. It differs whether that nitrogen is in the 5-membered ring or the 6-membered ring, whether you're doing purines or pyrimidines. When they bind to each other, and back in 1951, 53, 51, this was the insight that led Watson and Crick to discover the structure of DNA. Now, I know there's all the issue with them stealing Rosalind Franklin's X-ray crystallography photograph, photo 151. And as I said earlier in this semester, I'm not getting into that discussion. But what I will say is, having that, whether they got it honorably or not, having that helped confirm the suspicions that they grew from looking at the bases themselves. But it was looking at the bases that led them to propose the double helix. Because you might remember I mentioned in an earlier lecture that Linus Pauling had just predicted a structure of DNA that was 3 strands in a helix, with the phosphate backbones being internal and the bases sticking out into the just into the rest of the cell. Well, Watson and Crick, who had been specifically told by their bosses not to work on this problem, were noodling around with it in their boredom, in between other projects. And one of them, I believe it was Watson, cut out cardboard shapes. He drew the four bases on cardboard and then cut out the shapes that we see in front of us here, with the six-membered ring and the five-membered ring and all the various nitrogens and hydrogens and methyl groups sticking out. He drew those, and then he cut the cardboard to match the shape, and then he just started pushing them all together in various combinations and turns and twists, and trying two at a time, three at a time, trying all four. And what he eventually realized was, if he put them in specific combinations, A with T and C with G, and if he oriented them in a specific way with the sugar and phosphates sticking outward and the rest of the nucleotides pointing towards each other, he could get very specific hydrogen bonding lined up. And in this image on the right side, that's the little red hash marks between the H's and either the O's or the N's. Now you'll remember from chemistry that covalent bonds are very strong, but covalent bonds are very strong, but hydrogen bonds are not. They are fairly weak. So if you were to grab these two sugars and pull in opposite directions, you would not break any of the covalent bonds, like for instance between one of those unlabeled carbons and a nitrogen, but you could easily break the hydrogen bonds, because that's not a strong bond. So it was very easy to imagine a scenario in which these bases could be together under some circumstances and separated under others. And it was already known that DNA existed in the form of polymers, polymeric nucleotides, where they were connected from sugar to phosphate to sugar to phosphate to sugar to phosphate to sugar to phosphate, with the bases sticking off. And so if you could line up the nucleotides in a way that they hydrogen bonded to each other, then you could take one string of DNA and line it up next to another string of DNA, and they could attach like a weakly magnetic zipper. And what's more, the A's and the T's only form two hydrogen bonds with each other, seen at the bottom right, and the C's and the G's form three hydrogen bonds with each other, seen at the top right. And that meant that not only could two strands of DNA line up together and zip up, but that lining up and zipping had to be sequence specific. If you had a G on one side, you had to have a C on the other. If you had an A on one side, you had to have the T on the other. That was the only way that you would get the correct lineup of hydrogen bonds. So the very chemistry of these chemicals is what led to an understanding of their structure. And what happens if you try to line them up incorrectly? Well, for instance, if you try to line up an A and a C, you do not get good hydrogen bonding. In fact, if anything, you get obstruction. The two nitrogens that are lined up there, they're both accepting hydrogen bonds, meaning they have extra electrons that they want to share with a bound hydrogen. And at the top of the image, there's those two hydrogens butting up against each other. Those are both acceptors of hydrogen bonds. They both want to accept the electrons from the nitrogens, but they're not lined up to do so. So you cannot adequately line up an A and a C or a G and a T. And I will leave it to you to figure out for yourselves and maybe draw some things in your notes. Why can't you line up an A and an A, or a G and a G? Now, one thing I also want to point out, if you look to the image on the left, the G and the C that bind, that's between a purine, the G, and a pyrimidine, the C. The other binding pair is also between a purine, the A, and the pyrimidine, the T. In both cases, it is a matchup of one purine and one pyrimidine. This is not a coincidence. This is absolutely vital to the structure of the double helix. And we're going to be getting into the double helix pretty much the next slide. So let's move on. The double helix has a mystique about it. It is viewed almost as a sort of holy grail in the history of 20th century biology. It is the big event, even more so than the sequencing of the human genome in the 1980s and 90s. Part of that is the intrigue. Did Watson and Crick steal Rosalind Franklin's data? Was it given to them by Maurice Wilkins? How much of a role did misogyny play? How much was it poor communication? Rosalind Franklin had photograph 151 for over a year. She had multiple notes in her notebook on it and how she discarded it as being uninformative. Did her assistant give things to them? So it's got that sort of intrigue. And plus, there's the fact that it essentially launched Jim Watson into being a public figure in America. The combination of that and his own ego has kept him somewhat prominent, despite his racism and misogyny. There's all kinds of things that came out of this. Time magazine, interviews galore, books published. There's a lot of fame and American nostalgia built into the discovery of the double helix. So it's a big thing. But aside from all of that, the nature of the double helix, its physical structure, is responsible for all of life as we know it. Now, I'm not saying that it is the origin of life. I'm personally a believer in the RNA world hypothesis. But the physical three-dimensional chemical structure of the double helix is what makes life as we know it possible. So let's look at a double helix. And this here is a cartoon of our ribose or deoxyribose phosphate backbone. And if you look at the sort of blue shading behind it in the shape of an arrow, you will see that on the left side it's an arrow pointing downward, and on the right side it's an arrow pointing upward. And this is the 5' versus 3' ends. And these are labeled as such, as I've mentioned before, based on where the 5 carbon of the sugar ring is pointing versus the 3 carbon of the sugar ring. Now remember, chemists label the heck out of their chemicals, and every atom in some chemical compound is going to have a specific numbered position and a specific name. If you've taken any kind of organic chemistry, you are already well aware of this and familiar with it. And the way they number the bases, the carbons in a ribose, or a deoxyribose, numbers the carbons from 1 to 5. And the 5 carbon of a sugar, in this case, is where the phosphates attach. In general, when you make a nucleotide triphosphate, the 5 carbon is where the phosphate attaches. When you want to polymerize DNA into a chain, the first phosphate, the one closest to the sugar, that actually binds to the 3 carbon of a different base, of a different sugar. And that's how you get this chain. So if you look on the left side with the arrow pointing down, and pick any one of those little pentagon-shaped sugars. You will see pointing upwards and to the upwards and to the left is a phosphate, and downwards to the left is a phosphate. The one that's up and to the left is your 5' carbon, and the one that's down and to the left is your 3' carbon. So in each of these cases, the base initially came in with the phosphate that was up and to the left, and it wound up binding to the phosphate that is down and to the left. And that's how it got added to this DNA chain. And it is important to note that the strands of the double helix are pointing opposite directions. That is key because that lines up the sugars differently and it lines up the nucleotides differently. And that is what helps the double helix get its specific shape. You have to keep that in mind. That is like the number one key, most important part of all of biology, is the double helix has anti-parallel strands. Okay, so let's go back to this slide. Let's talk about DNA. And I've already talked about the structure of DNA a bunch. I am not going to require of you to memorize the structure of a cytosine, like where's the NH2, where's the double bond O. That kind of information is available all over the web, and at no point in your life is it going to be necessary that that is quickly available at the top of your head. And if you are at a point in your life where it is necessary, you will have been using this information so much that you will be remembering it anyway. However, it is important, hint, hint, like study for an exam important, it is important that you remember that the purines are A and G, and they have 2 rings, a 5 and a 6. The pyrimidines are C and T, and also U if you're talking about RNA, and they only have 1 6-membered ring. Whether or not they have O's or N's sticking off of them, I don't care that you know it. It is important that you know A and T bind with 2 hydrogen bonds, and G and C bind with 3 hydrogen bonds. And it is vitally important that you remember each DNA-base pairing involves 1 purine and 1 pyrimidine. Never 2 purines alone, never 2 pyrimidines alone, never other tuples of that. 2 purines and a pyrimidine, 5 purines and 4 pyrimidines. No. 1 purine, 1 pyrimidine, that's it. There is no spectrum of purine-pyrimidine identity. This is one of the few cases in biology where there is a binary dichotomy. So I started getting into this a few slides ago, but I want to illustrate it graphically for you here. How do the bases bind each other? So let's start off with this. We've got a triphosphate base here. We're going to move it over here, and what happens next is another triphosphate base is going to come moseying on in, and there are proteins that will be doing this. It's not just going to happen randomly on its own. It can happen randomly on its own, and that's probably how it happened the first time. But there are proteins involved in this making it happen. But a new DNA is going to come in here, and it's going to bind to one of the oxygens in that phosphate. In fact, what's going to happen is an oxygen on the sugar, an OH on that sugar, and that oxygen there are going to meet up, bind, and break a couple bonds, and a diphosphate is going to go flying off into the cell, and you're going to have a bond here, and it will generate a water. And so now, this is called a dinucleotide. We have 2 nucleotides attached to each other. And the thing I have circled in purple, where I have the O of the phosphate and the H of the other base, in real life that is just a single O bond. So one of those O's and the H are gone. Now we have a dinucleotide here, and what happens if another base comes floating along? Boom, we have a trinucleotide. So you see the closest phosphate stays attached, and the two terminal phosphates get cleaved off and go away. And those diphosphates can be used by any number of different processes in the cell. This is the basic unit structure of the double helix. I mean, that's all it is. This is life. This is all of life. It's a bond between a phosphate and a sugar. Okay. The reason I keep hammering home that you have one purine and one pyrimidine is, as I pointed out, the purines have 2 rings and the pyrimidines have 1. That means that they are unequal in size. So if you have a purine binding a purine with the 2 rings and the 2 rings, that's going to be a very wide pairing of nucleotides compared to a pyrimidine binding a pyrimidine, which is only 1 ring on each, and that's going to be a very narrow pairing of nucleotides. But if you have 1 purine and 1 pyrimidine, they're always going to be approximately exactly the same length. From one sugar to the next, the distance between them is uniform. That means whether you have a G or a C, whether you have a GC, an AT, a TA, or a CG, they are all the same width, that pairing of nucleotides. And that means when you form your double helix, the phosphate backbones are always going to be the same distance apart. That means the helix is going to be able to be a straight thing. It's not going to be turning and twisting and kinking and bending all over the place. Well, I mean, it can because it's long and fibrous, but it's not going to be doing so because it is out of shape. It is a well-made, straight structure. And it does not vary much based on the DNA sequence. Microscopically, to the level of sub-angstroms, it varies. But the sequence of the DNA, whether you have this base here or that base here, does not change the width of the double helix. There is something that does, and I'll get to that. There is something that does change with respect to the base sequence, and that's incredibly important, and I'll get to that in a few slides. But one thing I also want to point out here is the bases essentially stack horizontally one on top of the other. And in doing so, these the benzene rings and the imidazole rings, all of these various different rings, there's electrons being shared throughout this whole area. And by stacking them on top of each other in close proximity, the electrons actually get shared above bases as well. So from one base to its neighboring base to its neighboring base, vertically along the helix, along each strand of the helix, electrons are being shared. And that adds stability. That keeps it from sort of bending apart and breaking. Like if you've got one of those silicone molds for fancy shaped ice cubes, the way you pop the ice cube out is you twist and bend and it exposes the ice cube until it falls out. The electron sharing between bases prevents that kind of popping out of the nucleotides. So the bases stack one on top of the other. But they don't stack horizontally. They stack at an angle. And this is generally called a propeller twist. There's a slight angle, and the angle is dependent on the 5' to 3' orientation. So the one strand looks twisted in the opposite direction to the other strand. Now, there are exact, precise, and specific measurements of how far apart each base pair is, and how many degrees of rotation each base pair introduces to the double helix. Basically, what you're looking at is one full turn of the helix, 360 degrees, is about 10.5 bases. That makes each base roughly 36 degrees of rotation. Roughly, not exactly. All of this information is well characterized and publicly available, including the mass of each nucleotide and each nucleotide's length from sugar to hydrogen bond, or from one sugar to the next in a binding pair, or from one nucleotide to the next along a strand. All of that information is readily available and the kind of thing that might be nice to have on hand during an exam, like on a review sheet. And since it's so readily available everywhere, I don't even feel the need to provide it right here. One thing to keep in mind is a helix could be left-handed or right-handed, clockwise or counterclockwise, depending on which direction the strands are turning. DNA forms a right-handed double helix. And what we mean by right-handed is if you stick your hand out in front of you with your thumb pointing upwards, and you use your right hand for this, consider the underside of your hand in line with your pinky. That's where the 3' side of the strand is. And your thumb is where the 5' part of the strand is. So that's the direction of the strand of the double helix you're looking at. The direction that your fingers curve, that counterclockwise with respect to your thumb, that is the curve of each strand of the helix. That's the direction it goes as it winds up the helix. Now, if you look at your left hand and your right hand and you do some mental gymnastics and visualizations, you can understand how those are different from each other. But in general, when you're drawing DNA, any strand going up from left to right, you draw on top. Any strand going up from right to left, you draw underneath. And vice versa for strands going down. Now, I mentioned that the sequence of bases does not change the width of the helix, and that is absolutely correct. What the sequence of the bases does change is the shape of what's called the grooves. Now, if you look at the double helix, you have your phosphate backbones from each side of the helix, from each strand of DNA. And the space between one phosphate backbone and the other, as you look up and down along the helix, those are called your grooves. And there are two of them, the minor groove and the major groove. Now, these grooves are caused by the twist of the bond between the sugars and the nucleotides. That glycosidic bond that I mentioned. That's a 120-degree angle. And what you'll notice in the image here, the image in the center of the slide, the bases don't line up at the same angle as each other, depending on which sequence you have. So if you look, the bottom of that center image, you have a G on the left and a C on the right. And that looks like it's almost horizontal. It's maybe a little bit elevated on the G side. But if you look at the pairing two bases up with the C on the right, the C on the right and the G on the left, that no longer looks almost horizontal. That looks much more slanted. And similarly with the A and the T and the T and the A. One looks more horizontal and the other looks more slanted. Now, remember, these are still stacked, flat, slightly propeller-shaped as they go up the helix, turn, turn, turn. Remember, these are the bases in a double helix act as stairs in a spiral staircase. Okay, and in this case, the stairs are slightly angled. If you're standing on a staircase, the axis from your toe to your heel on the step, that is tilted. From left to right as you're standing on the step, that is not tilted. But what this difference in orientation is, it means that the step you're standing on in a spiral staircase is not going straight from one railing through the center to the other railing. The step goes from the railing to a place not quite centered, and then across again to the spot directly opposite you on the other railing. And so if you look in the center of a double helix, if you could see through the bases, it's not spokes of a wheel. There is no central singular axis that all the bases point through. Number one, because you have a pyrimidine on one side and a purine on the other, so it's going to be off-centered left to right. But also they're pointing at different angles, so it's going to be off-centered in other ways as well. So if you were to look at the end of a double helix right through the central axis, the bases would not line up in any kind of perfect symmetric pattern like that. They would be pointing off to each side at random kinks of angles. And that is what makes the grooves shaped differently. Because if we look at this center image, if we look at the very top of the image, there's a methyl group sticking off of that T, and then there's an O, an H, an N, and an H. Those are the bases, those are the atoms sticking off the top of the A and the T. That is what is showing as part of one of the grooves. And if you look at the top side of the bases directly below them, that G and the C, you've got the N, the O, the N, the N, the O, the H, the N, the H. Those are sticking off into the groove right next to the top pair. That's one groove. The other groove is what's on the bottom side of each base pairing. And that's easiest to see in the bottom set of images with the G and the C here, or rather the C and the G. So one of them is sticking off to one side, the other is sticking off to the other side. And because the sugars are turned, this glycosidic bond is turned at 120 degrees, one of the grooves is larger than the other. Because really, the helices are not lined up parallel to each other. Okay, I know I said that the strands are anti-parallel, because 5' to 3' goes one way on one strand, and 5' to 3' goes the other way on the other strand. That is correct. That is true. Now, imagine that I'm not talking about the strands in their 5' to 3' direction, but I'm just talking about them in their, if you look at a cross-section. As I've been saying, the bases don't point straight through the center. They point askance at each other. So, when you look at a cross-section of a double helix, the sugars are not 180 degrees across from each other. They are more like 120 or 150 degrees across from each other. And that means part of, if you think of it as a steering wheel or a clock, they are not at positions 9 and 3. They're at positions 10 and 2. And that means the area from 2 o'clock through 10 o'clock, the bottom 3/4 of the clock, or 2/3, that is your major groove. And the area from 10 o'clock to 2 o'clock, the top third of the clock, that is your minor groove. And this demonstrates that a little bit better. So this is that cross-section image that I'm trying to get you to think through. And the top half of each of these circles, or the top 2/3, is a darker gray, and the bottom third is a lighter gray. And that corresponds to your major groove and your minor groove. Now, what's really interesting about this drives most of cellular processes, because when proteins want to bind to DNA, it is very rare that they are, binding to an entire base, or they are binding to an entire nucleotide pair, or it's very rare that a base will be pulled out from its binding partner and be inserted into a protein like a key in a lock. No. Proteins are three-dimensional jigsaw puzzle pieces. They are three-dimensional Legos, and they are constantly looking for something that fits their grooves. All of protein binding is just, does your notch fit in my groove? Insert tab A into slot B. And when you look at the pairing of bases and which base is on which side, what's sticking out into that groove changes. And that's shown here with the red letters. So in these cases, A stands for acceptor and D stands for donor, and that's for hydrogen bonds, so where there's electrons versus where there's hydrogens. And the M stands for methyl group, which does not participate in the hydrogen bonds. Now, if you have a protein that has spots for hydrogen bonds in those same shapes, it'll fit. But a protein does not just bind to a single base pair. A protein that's going to come along and bind to the DNA, that's going to sit on a chunk of DNA like 6 to 20 bases long. So what you're looking at is like the inside of a tumbler lock, where, certain parts of the key have to be pushed up here and left down there and pushed up here. That's how it binds. You're not inserting a specific base into a slot. You're looking for a combination of about 60 different bumps and grooves in just the right orientation and shape. Now, the major groove, being larger, can accommodate proteins more easily than the minor groove, because the minor groove is smaller. The proteins don't fit very well. And on top of that, if you look at the minor groove side labeling in red letters, there's not a huge variety there. You've got 2 acceptors on the outside in all cases, and then you've basically got 1 donor on the inside. And it's just a minor variation in shape. Now remember, 10 bases is 1 full turn of the helix. So when a protein binds to DNA, most often it is binding in a 6 to 10 base long region, which is about 1 turn of the double helix, or a half a turn of the double helix. And that's how you get a protein that'll bind on one side, and you get another protein that binds on the opposite side, 5 bases over. And between the two of them, you now have 2 proteins bound on the DNA almost right on top of each other. And that can be the beginnings of a large multi-protein complex that can, for instance, replicate all your DNA. And it can bind to the DNA in a sequence-specific manner because of the pattern of bumps and grooves. Now, if you're having a hard time picturing this in your head, then what you can do is cut a bunch of circles out of a piece of paper, 6 or 7 of them. And cut one side of those circles to match these four these four red letters on each of these four images. You know, the major groove red letters. Cut the tops of each circle to match those. And then, keeping the circles slightly off kilter from each other so that each one points off, you know, a tenth of a turn from the next, put them all on top of each other. And then just look at the change in shape of these 10 paper circles. That is what a protein is binding to, that three-dimensional space. Those are your protein recognition sites. So we're going to be talking about restriction enzymes a little bit later in this chapter, actually. And we're going to be talking about transcription factors and initiation factors and translation start sites. All of these are regions where a protein binds to the DNA based on a sequence of bases. And this is how they do it. Okay, getting a little longer than I had wanted. Spent a little bit more time on that than I had hoped. We are now going to get into the Michelson-Stahl experiment, which has been described as the most beautiful experiment in biology. So this is one of those named experiments. We went through most of them in the very first lecture. You know, we had Hershey Chase and Avery McCarty McLeod. This is Michelson-Stahl, and this is really cool. So I presented this as part of a science journal club that I run for high school students. I presented this last year, and I'm going to give you a very shortened version of that presentation. I'm leaving out about 3/4 of the data and half of the explanation that I give. I just want to give you the basics, but I will tell you this. If you look at all the biggest names in molecular biology from the 1940s, 50s, and 60s, you got Jim Watson, Francis Crick, Sidney Brenner, Francois Jacob, Jacob Monod, Salvador Luria, Max Delbruck. They all have Nobel Prizes. Matthew Meselson has been robbed of the Nobel Prize twice. Now, again, I'm not getting into Rosalind Franklin. Whether or not we credit her with discovering the structure of DNA, the Nobel Prize is never given posthumously. And she was already dead by the time Watson and Crick and Wilkins got the prize for DNA. But Matthew Meselson is 90 years old this year. He's lives in California. He's been at Caltech for the past, 60 years. Jim Watson is still alive. He's 91 this year. Matthew Meselson deserves a Nobel Prize for 2 different experiments. He has yet to get one. Sidney Brenner deserved the Nobel Prize for 3 different experiments. He got only one of them, the third one. So let's talk about Meselson and Stahl and what they did. Okay, so this was late in the 1950s. DNA was really only known to be the source of genetic information for about 6 years. So this is 6 years after Hershey Chase, and it's 5 years after the double helix. So this is very early on. We know now that DNA is the source of genetic information. There's still some people that are holdouts for protein being the transforming principle. We have the double helix, and as Watson and Crick ended their landmark double helix paper, they said, it has not escaped our attention that the nature of this double helix lends itself to an easy replication mechanism. Sure, that's all fun and games to say, but no one actually knew what that mechanism was. I mean, it was obvious that you could pull apart the 2 strands of the helix, and each would be a template for the opposite strand, because you have to fill in the missing bases, and the bases are sequence specific. So if you have one strand, you have to know the sequence of the other. That's just how it works. So the obvious inference from this is when you replicate your DNA, You unzip the double helix, you put one strand on the left and one strand on the right, and you use each strand to make its complement, and now you have two double helices. That is the most obvious, simple solution, but it is not the only solution. So what are the others? Well, we have three possible methods. We have conservative replication, semi-conservative replication, and dispersive. And what are these? So semi-conservative is the model that is predicted by Watson and Crick. That is, you put one strand on the left, one strand on the right, each one gets a copy made on it, and then you have two. And when you want to do that, when you want to duplicate those, for each of them, you take one strand, put it on the left, one strand, put it on the right, and you make a duplicate. That is semi-conservative because the parental double helix gets split in half, and each, for lack of a better term, F1 double helix now has one parental strand and one new strand. So you have conserved one of the strands, but only one of the two, so you've semi-conserved. Well, another alternative is the conservative model, where the whole thing is conserved. So you start off with a double helix, and somehow you just make a carbon copy of it without destroying the original, and you still have the original. This was a very popular alternative hypothesis at the time, and it had a lot of proponents who were trying to say that, okay, well, you've got proteins that are going to bind to the double helix, and they're going to be able to recognize the shape because of the major groove, and so they'll be able to say, at this point in the double helix, we have an A and a T. So let's put an A and a T over here in this new double helix we're making, and we move on one base, and that's a C and a G. So let's add a C and a G to that A and a T, and boom, boom, boom. We're just going to read the original DNA and make a duplicate from what we've read. That's the conservative model. And then the dispersive model is everything goes everywhere willy-nilly. It's just all over the place. There is no pattern. The DNA just sort of gets broken up, chopped up, and duplicated who knows how. There wasn't really a lot of support for this model. Well, these are the early days of molecular biology and the early days of genetics. And one of the hallmarks of that is people sat down and just thought through logically, what would it take to prove this right versus this wrong, or that right versus that wrong? What would it actually take? And then they would design their experiments based on that, sort of a backwards logic. That is much more difficult to do these days because all the easy stuff's been answered, and the things we do now are a lot more of, well, what happens if I just tweak that? And what happens if I just take this away? And we learn a lot from that. And what they decided was they needed to create a new technique. So the technique they created is density gradient centrifugation, and the specific details of this aren't necessarily important, but they get this very heavy salt, cesium chloride, and they make a solution of that, and they spin it really fast for a very long time. They spin it 40,000 times per second for 2 days straight. Okay? That is a lot of spins. And it acts just like a carnival centrifuge. If you've ever been on one of those things, you're standing up and leaning against a sort of makeshift bed, and it spins so fast, you're pushed into the bed as if you were lying flat on the ground, even though you know you're vertical. It's the same thing with if you fill a bucket of water and you spin it in vertical circles. When the bucket's at the top of the circle, the water doesn't fall out because it's going so fast. So they created this idea of doing that with test tubes filled with cesium chloride. And when they do it, the cesium chloride actually shifts so that there's a little bit of it at the top, there's a middle amount in the middle of the test tube, and then most of it is at the bottom of the test tube. And if you look, depending on where you are from the top to the bottom of the test tube, there's a gradient of concentration. And that gradient is based on the density of the liquid. And the other thing they did was they grew bacteria in radioactive nitrogen. Or I should say, they know that bacteria make their amino acids from ammonia. That's the chemical process of how bacteria makes, nucleic acids, not amino acids. They make their nucleic acids from ammonia. So they put radioactive ammonia or radio-labeled ammonia in the bacteria culture. In this case, it's nitrogen, the 14 isotope, as opposed to the 15 isotope. And in some groups of bacteria, they gave it only ammonia with the N14 isotope, and in some batches of bacteria, they only gave it ammonia with the N15 isotope. Now, N15 and N14 weigh different amounts. Microscopically different, but when you have a large quantity of it, you can tell the difference. And lo and behold, if you have this really fast centrifuge and it sorts things based on the density, well, the density is going to be different because the nitrogen is not a different size, whether regarding which isotope it is, but it is a different mass. And mass per unit size is volume and density. So whether you have bacteria building their nucleic acids from N14 or bacteria building the nucleic acids from N15, they will centrifuge differently. And this is just a visual example of that. What it turns out is protein is the least dense of all of this. So if you centrifuge a mixture of DNA, protein, and RNA for a very long time, all of the RNA separates out and pellets at the bottom of the test tube, and all of the protein stays at the top, and the DNA forms a very, very tight band at a specific spot. Cesium chloride density gradient centrifugation is not used much anymore because it is unwieldy, it is long, and it is difficult, and it's very easy to mess up. Like, for instance, if you fumble the test tube in your hand after it's been centrifuging for 2 days straight, you've just lost all of that centrifugation. I did some of this when I was a research technician at a pharmaceutical company, and it is really satisfying to see that DNA band and to inject the side of a plastic test tube with a needle and extract that DNA band without getting any of the rest of it. That's very satisfying to do. Stressful, but satisfying. But so Meselson and Stahl came up with this concept of a density gradient centrifugation, and then they tested it, and lo and behold, it worked. So, for example, this is DNA in a test tube. Now, each of these horizontal bands is a photograph of a test tube that has been spun for the number of hours written on the left side. In this case, the photograph of the test tube is the test tube lying on its side with the top of the test tube towards the left and the bottom towards the right. And if you'll notice, the sort of narrowing bands of shade that become very tight in the middle by the time it's been spinning for 36 hours, that is where in the test tube DNA is located. When you first put the test tube in, it's all homogeneous, it's all shaken up, the DNA is everywhere. After you've spun it for about 2 hours, it's very slightly moved in from each of the edges, as some of the more dense cesium chloride drops to the bottom and some of the least dense water rises to the top and sort of squishes the DNA in between. And as a gradient of densities is formed along this axis, the DNA is restricted to that same area of its own buoyancy. So that line of black where the DNA is on the bottom 2 time points, that is where the cesium chloride has the same density as the DNA. And so it's buoyant at that level. So this is just a visualization of what we're actually looking at. In this case, there's no DNA or there's no RNA or protein shown in the image, just the DNA. I don't know why that was separated as a animation. Okay, so what they then did was they used UV absorption. They shined a UV light through the test tube. And UV light can be absorbed or refracted at different wavelengths. And we know the wavelength of DNA, and so they shown UV light of a specific wavelength through the test tube, and they saw that it was being absorbed at two specific points in the test tube, and this corresponded to where the DNA was located. So they were very visibly able to say, we have a band of DNA made from N14 labeled nitrogen ammonia, and we have a band of DNA from N15 labeled ammonia if we add bacteria from each of these two colonies together. So the next thing they did was they incubated bacteria in N15 ammonia for a very long time, for multiple generations, so that all of the DNA present in this bacteria was made from N15. All of it. Every single bit of DNA. And then they collected all this bacteria that has only N15 DNA, and they put it in a solution of only N14 ammonia. So any new nucleotides that get made, and therefore any new DNA strands that are going to get made, will only be N14 DNA. So in this case, if we think back to that conservative, semi-conservative model, the parental double helix is made only from N15. The second round of double helices, the sort of F1s, if you could think to call them that, any new DNA will be made with only N14. So you can make a prediction. If DNA replication is conservative, what you're going to see is this image. You are going to see your original double helix made only out of N15s, and you are going to see a new double helix made only out of N14s. You will have two distinct lines. And those two distinct lines will be specifically at the density levels for the N15 DNA and the N14 DNA. If you have semi-conservative DNA, then after one round of replication, well, before any rounds of replication, you will just have N15. After one round of replication, if it's semi-conservative, then you will have DNA that is half N15 and half N14. So you will have a single line between these two spots, between where you expect the 15 and the 14. And after a second full round of DNA replication, you will have something else entirely. You're going to have half of your DNA is going to be half and half, and half of your DNA is going to be N14 only. And so it's going to be shifting. It's not going to just be N14s and N15s. It's going to be a shifting balance. If you have disruptive DNA replication, you're going to start with just a band of N15, and then there's going to be some random mix of N14 and N15 after that in terms of ratios. And then that ratio is just going to further be confused and essentially be random in terms of how much N15 and how much N14 and where it's all going to go. So what do we see? These are different time points based on how many generations, so therefore how much replication has gone on. And if you look at the numbers on the right side, that tells you how many generations. So the 0 means there has been no DNA replication yet. You just have your N15s. So that is where the N15 band is. Now if you look at on the right side, if you look at the fourth row, you got 0, 0.3, 0.7, 1.0. That is after one complete generation. So you have now been growing the bacteria before spinning it. You have been growing it long enough for every bacterium in there to duplicate once. And then you spin that in your centrifuge. And if you look at that band is not directly underneath the N15. In fact, if you look above, you see a gradual shifting from the N15 band to a band slightly to its left, which in this case is slightly less dense. Now, if you follow that even further down, 1.1 generations, 1.5, now to 1.9, you have two bands of roughly equal proportions. One of them is the band that we just had that was slightly less than N15, and now we have another completely independent band. So, if we look at the next to last line, the next to last line says it took the 0 time point and the 1.9 time point and mixed them together. And you got three bands. Now, if you look straight up, those bands line up exactly with the band we see at the 0 time point, at the 1.0 time point, and the two bands we see at the 1.9 time point. And what are we looking at there? Well, the two bands I have highlighted in yellow are the N14 and the N15. So this is showing this, I'm just going to point this out. So this is all one round of DNA replication. This is a second round of DNA replication. And that is a third round of DNA replication. And so what we're looking at here is the bottom row in each of these three boxes. The bottom row in the top box is a semi-conservative hybrid. That is half N14, half N15. The bottom row in the red box is half N14. That's the one band is N14. And the other band are half N14, half N15. And then the bottom row of the blue box is mostly N14 and just a little bit of N15 left. So I will let you read through this slide on your own. This lecture is getting long and I want to move on. But this is a breakdown and explanation of what is found in each of these bands. And basically what the Besselson-Stahl experiment showed is DNA replication is semi-conservative. Each strand of DNA in a double helix remains whole, is not broken up, is not diced into pieces and used to make anything new, like in disruptive recombination. The double helix itself is not kept whole, as in conservative replication. The double helix is separated, and each strand is used as the template for the new strand. That is semi-conservative DNA replication. This should have gotten Messelson and Stahl Nobels. Messelson's still alive. He could still get it. I do not believe Stahl is. And this is just yet another illustration. And if you look at the bottom row, that's the 4.0 time point. That's the 4 generations. And you see all of the blue is N14 only. The red is N15. So just the sheer quantity of new N14 DNA outshines the N15. So the N15 band in the earlier images was not fainter because there was less N15 DNA there. It was fainter because for the amount of DNA present, the N15 was a much smaller proportion. It was 100% in the 0 time point, and here it's what? It's 2 strands out of 16. So it's 1/8 of the DNA. So it's 1/8 as bright. Okay, so we just learned that DNA replication is semi-conservative. Sure, okay, that's great. We've got a double helix. We got to separate it out and duplicate it. How? How are we going to do that? We do that with replication forks. We do not remove one entire strand from the other, leaving 2 single-stranded DNAs, and then start matching them up. What our cells do, and all cells that we know of, they create a small opening in the double helix by pulling apart the double strands in one small area, and then they insert a bunch of proteins onto each small single-stranded DNA region, and then they start initiating replication that way. So bacterial DNA is not arranged in long chromosomes like ours. Bacterial DNA exists as a circle. They don't have telomeres, they don't have ends of their DNA. Their DNA is just a circular double helix. Now, it is two strands. It's not like a Mobius of some kind. It is two strands, but they're wound around each other so that they physically cannot be separated without breaking the DNA. So what do we do? Well, and they locally separate the two strands, and then they attach to each side, basically covering each single-stranded section. And they start replicating it. And so in this image, the blue is your original DNA, and that's in the center. And it starts making new DNA, and that's the red that's being shown. There is a specific sequence of DNA called the origin, or the origin of replication. It's usually abbreviated ORI, ORI. And that is a place where the proteins involved in replication recognize the grooves of the major groove, the bumps and the nubbins, and they come in and they bind that. And they say, okay, this is where we're going to start. This is X marks the spot, let's dig here. And they start replicating at that point. Now, they can replicate only in one direction, or they can replicate in both directions. The catch is, replication always goes from the 5' to the 3', which means if they're going in one direction, they start from that origin, and they go off towards the 3' on one strand. And that's it. We will get into how the other strand replicates next time. If you remember this from previous bio courses, that's your Okazaki fragments. If it is a bidirectional origin, then one chunk of the DNA replication machinery goes onto one strand and says, oh boy, here we go, X marks the spot. And they shoot off towards the 3' side of that strand, replicating. More protein binds the other strand of DNA and says, all right, off we go. And they shoot off in the complete opposite direction because they're going to the 3' of that strand, because they're on opposite strands going opposite directions. Both are going from 5' to 3', but since the strands are anti-parallel, they're going different directions. You can have DNA replications going on more than once on a given strand of DNA. I want to point out the parts where the double helix separates out into its two individual strands, and then you have the new red DNA being generated there. That is called a replication fork because the helix forks. And essentially, if you have bidirectional replication, the origin of replication stays in one point, in one spot, and then the replication forks migrate away from it. If you have unidirectional, then the origin stays in one spot, one replication fork stays right there, and the other replication fork migrates away from it. If you have more than one initiation of replication on a chromosome, whether it's a bacterial circular chromosome or a eukaryotic linear chromosome, when replication forks bump into each other, it basically just creates one large area of replication. They merge. The two regions of replication merge to be one larger region. We are not going to get into torsion and topoisomerases here, but think about this before next lecture. If you take a rubber band and twist it a whole bunch, and you have this twisted rubber band, and you try and stick your fingers between some of the twists in the center and expand it outwards, the twists on either side of the center are going to get more twisted and curl. DNA is no different. The act of unwinding the DNA introduces twist and curl to DNA. You are creating torsional stress on this structure, and that stress has to be addressed. And we will talk about that in the future. There is another type of DNA replication besides the origin and the replication forks, and that is rolling circle replication. Now, the textbook image of this I felt was drastically confusing because there was nothing to show that there was a circle rolling. It seems like a tremendously wasted opportunity. So I inserted a couple of black hash marks here to show you that as DNA progresses, the circle of the chromosome rolls. In this case, rather than the two strands of the DNA being slightly separated, one of the strands is just flat out snipped and broken. And then the replication fork migrates from there. And that snip is shown in the second circle with the 5' P and the 3' OH showing. That is a break that is introduced in the DNA. And then replication starts. And it continues until the entire center strand is replicated. So that's rolling circle replication. That is used in bacteria. You just basically need to know that it exists and that it is different than other types of replication. I'm not going to spend much time on that. And then finally, this is our last slide of the day. You can see these replication bubbles. The image at the bottom of this screen is an electron micrograph of Drosophila DNA, and you can specifically see 5 different replication bubbles here. And those are eventually going to merge. And what happens when all of them merge? Well, when all of them merge, you're going to have one giant region of 2 double helices that are just sort of connected at the ends at replication forks moving outwards. And when those replication forks hit the ends of this linear chromosome, well, then you're just going to have 2 double helices. And you will have completely duplicated your DNA.